Good evening, everybody. I'm Professor Deborah Dunn-Walters, and I'm going to be your MC on this very special evening. I'd like to thank you all for attending what promises to be an enlightening, insightful lecture delivered by tonight's guest speaker, the inspirational Venki Ramakrishnan. So just very briefly, Venki's achievements in the field of structural biology are renowned. He won the Nobel Prize in 2009 for studies on the structure and function of the ribosome. He was knighted in the 2012 New Year's Honours List for services to molecular biology. I'm delighted you can be knighted for services to molecular biology. And he was the president of the Royal Society from 2015 to 2020. Although his honours are in molecular biology, Venki's PhD was actually in physics. And I think this is a lesson to all of us in the room who teach. He was initially put off biology because his biology teachers were not very inspirational. And fortunately for us, he realized later that biology and molecular biology was where his interests lay. But by that time, he had a lot of experience in physics and mathematics that he could bring to bear on questions in molecular biology and the problems of analyzing biological structures. So I don't know if many of you are familiar with the ribosome or not. It's the molecular structure that translates our genetic code into the proteins that make up the cells of our body. So literally, without the ribosome, we are nothing. It's extremely a critical organizational structure. With understanding of the ribosome, we are armed with information to help us understand fundamental processes governing life. So it's no wonder that the ribosome has been the focus of Venki's work over the years. His career choices have been led by scientific endeavor above considerations of geography and remuneration. It's not always been easy. As he says in his autobiography, universities did not know what to think of a physicist turned biologist using an esoteric technique. I like to think that times have changed and people are recognizing the value of interdisciplinary research. And certainly I'm very proud to belong to a university which values and encourages interdisciplinary science. Venki's work illustrates very clearly that there are important questions to be answered by novel approaches. So it gives me great pleasure now to hand over to our President and Vice-Chancellor, Professor Max Lu, to formally introduce Venki.
Thank you, Deborah, and good evening, ladies and gentlemen. It's a great pleasure to welcome you all, and what a packed room. And also for those online, I think we have a live stream. And to our distinguished guests, and the honored speaker, of course, Venki Ramakrishnan, to this year's Adams Sweeting Lecture. This lecture series, as we have been doing this for about 6 years, has a tradition of hearing from the eminent speakers at the forefront of science and technology. And tonight, we are so privileged, as Deborah already introduced, to be in the company of a man who is not only, in my view, exceptional scientist, of course, a Nobel Prize winner, but also an outstanding human being. So what he has accomplished in his chosen field in molecular biology is the result of his dedication and persistence that should be an inspiration to us all. I'm sure that he wouldn't mind me mentioning that, like many of us, many people in science, he encountered some rejections during the early years of his career. For example, when he applied for graduate school or medical school, indeed the first academic jobs when he was completing his PhD in Yale. And he did not get a single interview after, I don't know how many letters he wrote that he told his story at the Nobel Prize winning awarding ceremony. So many years later, I was fortunate to work with Venki and to see the same tenacity that he displayed in his career while working with him as a member of the PM's Council for Science Technology. By that time, he was not only the Nobel Prize winner, but also knighted, although whatever you do, don't call him sir. And Winky is famous for his humbleness and humility. Winky is also very thoughtful man, always collegial and pleasant to work with. Perhaps more importantly, a scientist who understands how science and technology can come together, and with other social science humanities to really make a difference, to do good for society. Cross-disciplinary collaboration was at the heart of his endeavor then, and also subsequently when he was the president of the Royal Society. He initiated many influential studies that had a huge impact in policy. So I recall Winky chairing a working group, which was I was a part of, focusing on broadening our education curriculum. And in this audience, you know that how our curriculum has become narrower and narrower. So in so doing, enhancing creativity, entrepreneurship among our students requires them to learn a bit broader in terms of their subjects in A-level. Winky was very aware of this issue, and then he set up this group within the PM's Council. I really admire his commitment to the idea that whatever our interest, our understanding of it, it is only going to be strengthened by close collaboration between the disciplines, philosophy, history, culture, and science and technology, and by examining what makes us human. Which brings us to the impressive breadth of tonight's lecture. This lecture series, of course, is named in honor of Professors Alf Adams and Professor Martin Sweeting, two Surrey pioneers whose achievements in science technology are simply extraordinary. So I'm delighted both of them are here tonight, and like Winky, both of them pushed the boundaries in their field, pushing the boundaries of knowledge through inquiry and innovation, and translating ideas into impactful outcomes. For example, Professor Alf Adams, by inventing the first generation strand layer quantum wave laser that is used in streaming to the internet, and Professor Martin Sweeting for pioneering modern satellite technologies. The impact of their world-changing discoveries and inventions are huge. So as Deborah alluded to, Winky's lifelong interest in ribosome only began after an unusual transition from physics to biology, but his work's impact is much wide. It's huge and widely into antibiotics, cancer, memory, and viral infection. So with that, I'd like now to take this opportunity to salute Winky and to now invite him to take the podium to give us a lecture. Thank you very much, Winky.
Thank you. Thank you all for coming, and it's certainly an indication that COVID has at least subsided, if not gone. So I want to talk to you about why seeing things is important and what it has meant in the field of chemistry and biology. So why is it important to see things? Well, imagine if you were some alien who had sort of hovered about the Earth, and it was the first time you saw a car, okay? You'd have no idea what this car actually was. You'd know that it went around, and well, in the US it would go in straight lines and turn at right angles, but you know, I don't know about Surrey. But anyway, you'd see it moving around, and it would stop, and then you know, little objects would get out of it, and when the objects had gotten out of it, it would stop, and it wouldn't move again until those objects had re-entered it. And so you'd wonder what it was like. And then if you got closer, you would see that, oh, well, it seems to have a certain shape, and it has wheels, but you still had no idea what it would look like. Then you probe a little further, and you look at it more, and you see that, oh, it's got 4 wheels and it's got something here that generates power, and this thing here, we have no idea what it does, but it seems to be important because all these objects seem to have it. It's only when you look at it in detail that you see that this thing that generates power actually is made up of pistons that, take up gasoline, and, there's a spark, and there's combustion, and it pushes down on a piston that generates force and turns the wheels, and this little thing here is what makes the wheels turn, and so on. Suddenly, by looking at the details, you've understood what this machine actually does, what it looks like, and what it does. And it's the same thing with almost everything, even in astronomy. When Galileo looked at the moons of Jupiter, suddenly planetary motion became, an idea. It greatly strengthened the heliocentric theory. Kepler's laws were made simply by observing how planets moved, and that then led to Newton's laws of gravitation. So it's been dramatically important, even in astronomy. Now, at the other end, if you go to zoology, if you go from A to Z, then this is an interesting picture, because it's a picture of Andreas Vesalius' book on anatomy. Now, Vesalius lived, I believe, in the 1500s, and he was interested in what the organization of the body was. Now, people had dissected animals and made conclusions from animals that didn't actually work for human beings, and that was the case for centuries. And most religions, in fact, forbade you from, dissecting bodies. In fact, the idea was that, you should, if somebody died, you should bury it or cremate it as quickly as possible. And what Vesalius did was to actually go to graveyards and steal corpses and dissect them. Okay, and as a result, he changed our understanding of anatomy, and anatomy has never been the same since. Now, he was able to see all the layout of the muscles, the various organs and tissues. But if you wanted to look at what was actually inside the tissues, what were these tissues made of, then you needed to look at it in more detail. And that then asks, how do you look at things in detail? This means, looking at very, very small features, and you're trying to distinguish them. Well, the way you would do that is you would take an object and put a lens in front of it. And what the lens will do is collect scattered rays from the object, and then project that into a magnified image. And then you can look at the magnified image and see details. In fact, all of you now are looking at scattered rays from the screen, and the lens in your eye is concentrating them, recombining them into an image on your retina, and that's how you're able to see. Okay? So this idea was used by this man, Antony van Leeuwenhoek, who was a Dutch linen merchant, and he was clearly interested in looking at the fine details of the fabric that he was buying and selling, to look for defects or look for quality. And this led him to develop very powerful magnifying glasses. And in fact, we call one of these, we think of it as the first real microscope, because his magnifying glasses, this microscope only consists of one lens, but he used to make his own lenses, and his lenses were better than anybody else's at the time. And they were so powerful, and he was naturally curious, and so he would look at all sorts of things. For example, when he looked at pond water, he found little microscopic things swimming around in the pond water that he couldn't see. And he called them animalcules, but actually they're what we call microbes today. And so that was the foundation of the entire field of microbiology. In fact, he did things like scrape the scum off his teeth and look at them, and he was shocked to find, it's crawling with creatures. Okay, so that was van Leeuwenhoek, and he, interestingly, in a sort of European collaboration. He communicated all his work through the philosophical transactions of the Royal Society, or actually as letters to the Royal Society. And at the Royal Society, around that time, there was Robert Hooke. And Robert Hooke was a polymath. You know, he was a physicist, a biologist. He was all sorts of things. You didn't categorize people in those days. And one of the things he noticed was when he looked at tissues of cork, he found that they were organized in these cells. And so that's where the word cell comes from it. And today we know that the cell is the unit of all life. Okay, all living creatures are organized as cells, with the exception of viruses, which have to invade cells in order to survive, in order to replicate. And so you can see how just observing things has transformed, transformed astronomy and physics. It's transformed anatomy, and then modern biology. And using microscopes, which were more and more powerful, people began to understand the organization of cells. For example, they realized cells have nuclei, and inside them there were chromosomes that would then, and during cell division, these chromosomes would separate, as shown here. And so people understood quite a lot about the organization of the cell. But when you ask, what are these chromosomes made of, or what are these structures in the cell made of, then you had to try and understand what these molecules inside cells look like. And before I get on to molecules, I forgot this. This is a very interesting picture. This is looking at neurons in the brain, in brain tissue, by staining them with a dark stain called the Golgi stain. And this was done by a Spanish scientist, Ramon y Cajal. Now, before this Golgi stain, when people had looked at brain tissue, it was just this big tangle of fibers. And people couldn't really make any sense of it. And what Ramon y Cajal realized was actually the cells in brains were not like typical cells, which were sort of more or less round or roundish. Rather, they had a cell body from which there were long extensions. And what he also realized was these extensions would somehow connect with extensions from other cells. And so he realized that the brain was really a highly interacting set of cells, where it would send off these long spindle-like extensions to talk to other cells. That laid the foundation of neuroscience. Okay, and everything we know about neurobiology comes from that work by Ramon y Cajal. Now, in an odd twist, Ramon y Cajal shared the Nobel Prize with Golgi, but Golgi didn't like Cajal's interpretation of the brain, using Golgi's own stain, I should add. And so he spent his entire Nobel lecture attacking Cajal. Okay? It must be the first, possibly the only time in the history, although I can think of a couple of economics Nobel Prizes where it was shared by two people who completely had opposite ideas, but that's economics. So anyway, that was Ramon y Cajal. So now, if you want to understand what's inside these cells and what's inside them, you have to know something about molecules. And the amazing thing is that scientists had figured out what molecules were and even their structure without ever having seen one. So in the 18th and 19th century, chemists had figured out that matter consists of atoms, and that atoms come together in very regular arrangements to form molecules. And this is an amazing intellectual feat. And if you want to sort of learn about the history of how we came to that understanding, this is an excellent book called Chasing the Molecule. But no one had actually seen a molecule because molecules were too small to see by a light microscope. Because people knew that simply by calculations from Avogadro's number. They knew what molecular weights were, so they knew roughly what the size of molecules were. and it was much, much smaller than the wavelength of light. And so you couldn't use, there's a law in physics that you can't resolve things which are much smaller than the wavelength of the light you're using. So in 1901, the first Nobel Prize was awarded to, no, it wasn't the first, but one of the early ones was awarded to Röntgen for the discovery of X-rays. But no one knew what X-rays were. They just knew them as a radiation that could penetrate objects. It was when Max von Laue, a German physicist, took a source of X-rays and hit a crystal with it, and then looked at what happened when it hit an X-ray, that we began to understand both something about crystals and something about X-rays. So from this, he realized that people knew crystals were regular arrays of atoms or molecules. And what von Laue realized is that somehow this regular array was causing interference of the X-rays. So from that, he actually showed that X-rays were actually waves. Of course, today physicists believe that they're both waves and particles in this particle-wave duality. But in any case, they have wave-like properties, and these spots were the result of interference between these regular arrays of atoms that make up the crystal. Now, he had absolutely the right idea, but his analysis turned out to be wrong. And the fact that it was wrong was actually noticed by this man, Lawrence Bragg, who was at the time a PhD student in Cambridge. And what Bragg realized is what must be happening is a result of interference between regular planes of the crystal. Now, before I can explain what that means, you have to ask, how do waves actually add up? So if you have two waves that have to combine with each other, what would the result be? What would the combined wave look like? Well, what you would do is at each point, you would take the height of the wave. So if you draw a zero line here, here's where it would be highest, here's where it would be lowest. And at each point, you would combine the heights, and the net result would be this combined wave. Now, imagine if the heights, the peaks of the two waves coincide and the troughs also coincide, then you would get the maximum addition. But if you have a peak of one wave coinciding with the trough of another wave, then they would absolutely cancel out each other, and you would have essentially no intensity at all. So you can see that in this. Well, I'm sorry, this movie isn't working, but I've explained it to you. Okay. So anyway, if these waves were then to move around, then what would happen is that you would get maximum intensity when the peaks match up. So what Bragg realized is that in the crystal, you can think of planes of molecules or atoms, and then what will happen is that X-rays will bounce off those planes and cause spots only in certain directions. And this is actually taken from Bragg's paper, which was published in the Philosophical Transactions of the Cambridge Philosophical Society. And it was in 1912, so this was actually just over 100 years ago. Now, if you look at what's happening, you can see that X-rays might bounce off one plane, and another X-ray might bounce off an adjacent plane. Now, this X-ray has to travel a little further than this X-ray, okay, because it's gone one plane down. And the extra distance is shown here. It's like if this is L, then it's 2L. That would be the extra distance it travels. And the extra distance can be related to the angle of the scattering and the distance between the planes by this number 2d sine theta. Now, what Bragg realized is if this extra distance is a whole number of wavelengths, then what will happen is, even though it's gotten behind, if it goes behind exactly one wavelength, the peaks will still match up, okay? And when they add up, they'll add up to a maximal value. And if you look at crystals, you can think of crystals as regular arrangements of atoms, but you can generate planes in an arbitrary way. For example, this arrangement of atoms you can think of as a set of planes going like that, and this would satisfy this Bragg condition where it matches up a whole number of wavelengths for a particular angle. But the same arrangement of atoms can be thought of as planes in a completely different direction, okay? But the difference is that the distance between the planes will be different, and now it'll be a different angle that will satisfy that condition for the Bragg condition. And so using that, Bragg was able to explain how you would get spots only in certain directions, depending on which plane was resulting in the scattering and giving rise to this Bragg condition, which allowed the waves to add up. So based on that, he decided to tackle a very simple substance, which was common salt. Now, common salt, everybody knows, is sodium chloride. It's a sodium atom and a chlorine atom, and chemists had figured this out. But when Bragg determined its structure, what he found was that there was no sodium chloride molecule. Rather, it was this sort of checkerboard of a sort of three-dimensional alternating checkerboard of sodium and chloride ions, okay? And for those of you who are solid-state physicists, this is an interpenetrating, face-centered cubic lattice, okay? But basically, you can think of it as an alternating checkerboard of sodium and chloride ions. Now, chemists didn't like this. Remember, Bragg at the time was still a PhD student, and and in physics at that. He wasn't a chemist. And in those days, if you think we're siloed today, imagine in the early 20th century, physics departments and chemistry departments, they hardly ever talked to each other. So when Bragg eventually established himself and published an article, this was a few years later, a chemist, Henry Armstrong, who was a professor of chemistry at UCL, wrote a vitriolic letter to Nature in which he said, "Some books are lies from end to end," says Burns, "and scientific speculation would seem to be on the way to this state." He says, on page 414, Professor Bragg asserts, "In sodium chloride, there appears to be no molecules represented by NaCl. The equality in number of sodium and chlorine atoms is arrived at by a chessboard pattern. It is a result of geometry and not a pairing off of atoms." He's quoting Bragg. And then he goes on to say, "This statement is more than repugnant to common sense. It's absurd to the nth degree and not chemical cricket." Clearly, he was an English gentleman, and that was the worst insult you could make to somebody. And then, "Chemistry is neither chess nor geometry, whatever X-ray physics may be." So that was the initial reaction of the chemists. But of course, only 3 years after Bragg's discovery, he shared the Nobel Prize with his father and remains the youngest science Nobel laureate, having received it at the age of 25. Now, if you look, the way Bragg did it was he would guess at the structure. He was working at relatively simple molecules, like sodium chloride, and he would guess at the structure, and then based on that, he would calculate what the X-ray scattering should be, and then see if it matched. And if it didn't match, he would, make other guesses until it matched. That's okay for small molecules. But even if you have a handful of atoms, that starts getting to be difficult. And so there was another method that was used, and that is to look at, you have to think about how lenses work. As I mentioned to you, what a lens does is it takes scattered rays and recombines them into an image. Now, the point is, whether you have a lens or not, the information to make that image is there in the scattered rays. For example, I see a couple of empty seats there in the second row, and there's nobody there, but the scattered rays from the screen are there, and there's information there. So if someone were to come and sit there, they'd be able to see the screen, right? So you don't need the lens. All the lens is doing is recombining the rays to make the image. Now, in principle, you could take the scattered rays and measure them, those spots that are generated by crystal, and combine them to make that image. And that would then allow you to regenerate the image of the molecule. And that's sort of shown here. So, for example, you would take the X-rays, you would take the scattered rays and measure them, and then in a computer, you would do what a lens does, which is recombining the rays. And what a lens does, actually, is calculate a Fourier transform of the scattered rays. And that's what you would do in a computer. Now, there's a problem with that, and that is when you measure the intensity of those scattered spots, you're only measuring one of the properties of the waves, which is you're measuring how high the peak is. In other words, what the amplitude of the wave is. The more the amplitude, the darker the spot, or the more intense the spot. But what you need when you want to recombine all of the waves corresponding to the different spots is you need to know how far advanced the peak is of each spot relative to the others. Okay? In other words, you need to know this angle, this phase angle phi, which connects this peak with that peak. And that's the part that you don't know. Okay? And that's what's called the crystallographic phase problem, and it was figured out over a period of decades, first for small molecules and then for large molecules. And one of the ways you can do it is, if you have in your structure a particularly dense atom, which is much heavier, that is, has more electrons than lighter atoms, then what you can do is you can use methods to figure out where that atom is. Then you can calculate a structure assuming you only have that one atom. And when you calculate the structure, you'll get amplitudes, the heights of the waves, but you'll also get the phases for all the spots because you've done the calculation. What you then do is use the actual measured intensities with the calculated phases. This gets you slightly closer to the truth because the intensities are the ones you actually measured that are correct. The phases are not actually accurate because you've only put in one of the atoms among the many atoms in your structure. But there's still some information there. When you do that combination, what you get is a strong peak for the atom you actually put in, but you get weaker peaks for the atoms that you didn't put in because you now got closer to the truth. And you can sort of bootstrap your way. And what you do is then you put in atoms where you see them in your calculation, and then do the recalculation. And that's a better structure. And then you slowly get to a better and better structure until you eventually solve it. Now, the person who really used this method to its utmost, one of the people who did that was Dorothy Hodgkin, who got her PhD from Cambridge, where she learned how to do crystallography, but then spent most of her life in Oxford. I believe she's the only person who's had 2 stamps, postage stamps, named after them. And this shows the image she generated for penicillin. She had an exceptional knack for studying really important molecules, and which is why she was awarded the Nobel Prize in 1964. Now, penicillin has this unusual 4-membered ring made of 3 carbons and a nitrogen, and it's called the beta-lactam ring. And when chemists saw it, they hated it. just like when chemists saw Bragg's structure, they hated it, okay? And John Conforth, who was an organic chemist, looked at that structure and he said, if that structure is right, I'll give up science and start growing mushrooms. And well, as it turned out, the structure was right, but Conforth didn't keep his word. He kept on doing science and won his own Nobel Prize for synthesis of steroids, I think in 1975, about a decade later. Anyway, one of the things that Dorothy Hodgkin did was solve the structure of vitamin B12, which has several hundred atoms. This was a tour de force for its time. So luckily, it has a cobalt atom, which is much denser than the carbon, nitrogen, and oxygen atoms surrounding it. So she could locate the cobalt, and then doing that initial calculation, she could locate other atoms, and then she would put those in, and then she would finally get the entire structure. And this is taken from her Nobel lecture. This is the entire structure of vitamin B12, determined entirely by crystallography, and then subsequently it was verified by chemical methods. So, you can solve structures up to a few hundred atoms using this sort of method. But what about when you have several thousand atoms? For example, proteins are molecules which have several thousand atoms. Well, that problem was solved as a result of an idea that Max Perutz had. Max Perutz is also the person who founded the institute where I work, the MRC Lab of Molecular Biology. And what he realized was that if you took a crystal containing a protein and you added a heavy atom to it, like mercury or gold, it would, and if that atom could sit in specific pockets in the protein, then you could have a crystal of the protein alone and a crystal of the protein with the heavy atom. And the two of them should show slight differences in intensity. And what he figured out was from that difference in intensity, you could calculate where that heavy atom was in the crystal. And if you knew that, you could then figure out what the phases for the reflections were and calculate your structure. And just to show you how small these intensity differences are, this is a photograph of the protein alone, and this is a protein with the mercury. And you can see that they look almost identical. But if you look very carefully, just above this red line, you can see a group of 4 reflections, and they're roughly equal in intensity. Now you go to the same reflections here, and what you see is the second one is much weaker than the others. And that change is a result of adding the heavy atom, okay? You wouldn't be able to see that unless you did a very careful comparison. But with computers, you can do that. You can scale them and look for the differences and then calculate the differences. So using that, Max Perutz and his student and then colleague, John Kendrew, both shared the Nobel Prize in 1962. Kendrew had very, very cleverly chosen a more tractable molecule called myoglobin, which was 4 times smaller than hemoglobin, which was Max Perutz's molecule. So at the time of the Nobel Prize, Kendrew had already solved the atomic structure of myoglobin, which is what convinced the Nobel Committee that this was actually working and it was worth awarding. But Max had still not solved hemoglobin, although he went on to solve it a few years later. So it's shown really as this sort of balsa wood cutout model. Now, if you want to look at much larger molecules, you need very powerful sources of X-rays. A lab X-ray source isn't strong enough to give sufficient signal from crystals of extremely large molecules because the diffraction, the spots from those molecules, are much weaker. Generally, the larger the molecule, the harder it is to make crystals, the fewer molecules there will be in a crystal of a certain size, and so your spots will be weaker. And so what we generally use are these very powerful accelerators called synchrotrons, which make electrons whiz around very close to the speed of light, and in doing so they emit very powerful beams of X-rays. And I should point out that Britain has a premier synchrotron at the Diamond synchrotron, which is at Didcot, not very far from here. It's close to Oxford. And if you want to use synchrotrons, there is a problem. If you have very, very powerful sources of X-rays, very intense X-ray beams, they will damage the crystals that you have. And so the crystal will be fried before you can even collect data from it. And that problem was solved by a number of people who went on to show that if you cooled crystals to close to liquid nitrogen temperatures, then you would slow down that radiation damage and you would allow data to be collected from these crystals. And this is just to show you what an inside of a synchrotron looks like. Here's a pipe that brings the intense source of X-rays to the crystal, which is here. This pipe is actually a tube that provides very cold gaseous nitrogen from boiling off liquid nitrogen to keep that crystal very cold. And this is an area detector. They used to be CCD detectors, but they're now called direct pixel array detectors, which calculate which measure the intensity of the X-ray spots from the crystal. All right, so I should say one other thing about synchrotrons, and that is, just like when you add heavy atoms, your intensities change slightly, and those intensity differences allow you to calculate the structure. In synchrotrons use something called anomalous scattering. So when a crystal is symmetric, which many crystals are, then you can have orientations where the pattern is also symmetric. So for example, on this X-ray picture, the left and right side of the patterns are identical. They're essentially mirror images of each other. And what Byford showed is if you have atoms with certain special properties where they absorb X-rays and then re-emit them, then this mirror symmetry is broken, so the intensity is no longer identical in the left and right side. There's small differences. And if you measure those differences accurately, you can do the calculation to get you your structure. And the advantage of synchrotrons is, unlike a lab X-ray source where electrons hit a copper, rotating copper anode, and then give off X-rays of a particular wavelength, with synchrotrons, you can tune the wavelength of X-rays that you want. And so you can tune it so that those anomalous scattering properties of special atoms are maximized. And this is how very, very large structures were solved, by using this property of synchrotrons to get phase information. Now, when you solve a structure, you don't get the position of the atoms. You effectively, I should probably, what happens is you have to look around and recognize features. For example, you can see a ridge here and another ridge here, and you can see regular bumps here, etc. And if you know something about what you're doing, what you can do is you can interpret that in terms of chemical structure. So for example, that pair of ridges was actually two strands of RNA that are wrapped around each other to form a double helix. And so suddenly you've interpreted the image in atomic terms. And what you have to do is you've now interpreted just one part of the image, and you keep doing that until you effectively get the entire rest of the image. And when you've interpret the entire image in molecular terms, in atomic terms, then you've effectively solved the structure. And this is exactly how the ribosome, which you heard about in the introduction, was solved. This is a molecular complex which has 500,000 atoms. And so remember, crystallography started with sodium chloride with 2 atoms. And in sort of 2000, you could solve things with almost 500,000 atoms using advances like synchrotrons and computers and X-ray detectors and liquid nitrogen sources to keep crystals cold, etc. And so that's just showing you the power of crystallography. Now, not everything crystallizes. In fact, crystallization of large complexes can take years. One of the reasons that Ada Yonath received the Nobel Prize for the ribosome was she was the first person to show that they would crystallize. And so what could you do if you don't have crystals? Well, I told you couldn't use light microscopes to look at molecules because the wavelength of ordinary light is far too large. But you could use electrons. Electrons can also be focused. For example, if you have a ring of electric current, that ring essentially acts as a lens to deflect electrons. And so you can sort of focus electrons with these electromagnetic lenses, just like you could do with a glass lens and light. And that was first realized by this man, Ernst Ruska, who invented the first electron microscope in 1933, and this is what that first microscope looked like. His timing was a bit unfortunate because soon after that, in 1933, Hitler had already assumed power, and a few years later, World War II broke out. And after the war, the use of electron microscopes became commonplace, and people simply forgot that they'd actually been invented by someone. And it's only 50 years later that somebody realized that, hey, we've not awarded a Nobel Prize for this invention that everybody is using in everything from biology to material science. And so he received the Nobel Prize 53 years after his discovery. And note that he died 2 years after that. And so if they had waited another 2 years, he would never have got the Nobel Prize. So one lesson, those of you who are aspiring to a Nobel Prize, is to stay healthy and try and live as long as you can. Okay. So anyway, so moving along, how could you get three-dimensional structures from an electron microscope? Because you get essentially a flat picture. You're not getting a three-dimensional picture when you focus your electron beam on a sample. Well, what you get when you take an electron micrograph is essentially a two-dimensional projection of the object. So I don't know so for example, if you look at the shadow of my hand, it looks very different if I have this projection compared to that projection, right? And so that's what you would get. You'd get a series of two-dimensional projections. But what Aaron Klug realized is that you can take these two-dimensional projections and recombine them in the computer to generate back the three-dimensional object that gave rise to those projections. But for that, you would need to look at it from, lots of all possible angles to be able to reconstruct that three-dimensional object. And for this, Aaron received the Nobel Prize in 1982. And Aaron, by the way, was the director of the lab where I worked. And in fact, he was the director when I went there first on sabbatical. And interestingly, he also became the president of the Royal Society later. Now, this technique has recently become very, very powerful and used to determine the atomic structure of biological molecules, for which these people shared the Nobel Prize. Now, Richard Henderson, who was also a director of the lab, in fact, he's the one who gave me my job, published a very important paper in 1995 saying that in principle, the signal from an electron micrograph had enough information that if you averaged over a number of particles, you should be able to calculate an atomic resolution structure. And over the years, he went on to drive improvements in the microscope and in the detectors in order to help make that happen. This man, Jacques Dubochet, he showed that if you take samples in aqueous environments, all biological molecules exist in aqueous environments unless they're in lipid membranes, and even membranes exist in the context of an aqueous environment. What he showed was you could take this, make a very thin layer of it, and flash cool it. It'd cool it so fast that the water wouldn't have time to freeze into ice. And so effectively, this would be vitrified water. It would be like a glass. So it's not what physicists would think of as frozen, because it's not a solid. So it's really what's a cooled liquid, a glassy liquid. And what Joachim Frank realized was, instead of taking a sample and then rotating it in the electron beams in order to get different views, what you would have in this, if you took a solution of molecules and then made this flat, thin layer and froze it like Dubochet, was you would trap molecules in all different orientations. And I don't know if I have a slide here. So if you took a vitreous layer of ice, you would have this molecule in all different orientations. Each of them would give us its own projection under the electron micrograph. And what you would have to do is you would have to figure out which orientation gave rise to that projection. And if you could do that, and there are now clever algorithms that allow you to do that, then you would be able to reconstruct the three-dimensional image of the molecule. Now, I think I have a movie that shows you this. So that's the electron micrograph, and you can see that's the object that is giving rise to different projection views. So if you have a projection, you have to figure out which angle of the object gave rise to that particular projection. And if you have a different projection, then it'll map at a different position on the sphere surrounding the molecule. And if you have lots and lots of those projections, then you've effectively covered as much of the sphere as you can, and in the end, you will end up with a three-dimensional image of the object. And this is how the technique of cryo-EM works, and it's completely revolutionized structural biology. So for example, one of the kinds of proteins that was very, very difficult to do by crystallography is the class of proteins called membrane proteins. These are not soluble in water. They have to be solubilized in detergents, and even then they don't crystallize usually, and if they crystallize, they're not very good. And it's very, very hard. But now with cryo-EM, people have been able to solve huge complexes. So this is a molecular complex that's responsible for generating ATP. This part of the molecule rotates like a turbine, just like a turbine rotates to produce electricity. This molecule rotates to produce ATP from ADP, and for which Paul Boyer and John Walker also was at the LMB, shared the 1997 Nobel Prize. And this is a molecule that's responsible for pumping protons across the membrane of mitochondria. And it's actually that proton gradient that drives this turbine to make ATP. So they're sort of related mitochondrial complexes. And this is a pore that the anthrax bacillus can use to insert into your membrane and cause us problems. And so you can see there are very, very large important complexes now being solved by cryo-EM. Now I just want to show you a movie let's hope this works. This is a vesicle, and in our cells, large factors, large parts of our cell are actively transported from one part of the cell to the other. They could be molecules, or they could be entire organelles. Imagine, for example, that one of our axons can be 1 meter long, okay? And things can't diffuse through the axon. It would take it would take days or even weeks to get from where they're made to the synapse where they're needed, So cells actually have transporter molecules that will actively transport cargo from one part of the cell to another. And one of those transporting molecules is called dynein, and that's been studied by my colleague Andrew Carter, who made this movie. So here's this vesicle, and you can see there are these long filaments that get attached to the vesicle, and then eventually what happens is a pair of molecules called dynein and dynactin connect to that cargo. And then what dynein does is it finds tracks, which are microtubules, which are assembled by the cell, and then walks along the microtubule in order to transport it. Now, while it's doing this, it's actually dragging that enormous vesicle along. So it's a fairly small molecule in comparison to the enormous vesicle. And this is how, for example, even viruses or organelles like mitochondria are all transported through the cell. And so this is all done by looking at snapshots of the structure at different stages so you can see those conformational changes. It looks scary. It looks like an alien creature with feet walking along. But they're actually just parts of a molecule that undergo changes as it grabs onto the microtubule and releases and then contacts it at a different place. Now, you can also actually look at entire cells and look at molecules in cells as they are without even isolating them. This is a method called cryo-electron tomography. And this is a movie made by Tanmay Bharat, who's a new colleague of mine at the LMB. And it shows a bacterium which has a cell body here with a stalk, which you can barely see, but it'll become clearer. And using this technique, you can actually look at the molecular organization of the stalk, which is what helps this Caulobacter move. So you can see this is a three-dimensional view of the object, and he's now going to focus in on the stalk. And when you look at it very carefully, you see it's a regular organization of these molecules which make up the stalk. And if you zoom in on it, you can see exactly what that stalk is made of and what the molecules, what the molecular organization of the stalk is. Now remember, this is done without isolating the molecules. You're simply looking at entire biological specimens, and you can see exactly how the stalk is organized by doing this cryo-electron tomography. So we've gone a long way at looking at very simple molecules like common salt over 100 years ago to incredibly complex molecules, some of which you've isolated, but some of which you've never even isolated. They're inside the cell exactly as they would be present. So I'd like to conclude there, and thank you very much for your attention.
Thank you very much. All PhD students in the audience remember about Lawrence Bragg, right? So we're going to have a question and answer session now. And we do have people online, and so if in the audience you would like to ask a question, and please do, can you wait until the microphone arrives? There'll be someone running down here and someone running down there with the microphones. I will also look here because people online, you are also very welcome to ask questions, and they will appear here so that I can ask them. So if you would like to take a seat, thank you. Thank you very much for that. That was absolutely fascinating. Do we have anybody in the audience here who would like to ask a question at this point? There's a hand up in the middle there. Thank you.
Yeah, and if you could speak a bit loudly and clearly, because I wear hearing aids and I'm not great at hearing.
Hi, hello. I found this really interesting, and I was wondering if with the release of AlphaFold predictions of all the proteins, if you think this is the end for crystallography or not.
So I personally am a big fan of AlphaFold. I think there's still cases where it doesn't get it quite right, but what's astonishing is how it gets it right so often. And AlphaFold is now being used extensively by people who do electron microscopy or crystallography in order to interpret those images. Because instead of having to sort of painstakingly guess residue by residue, what the structure is, you can look at the AlphaFold prediction and you can instantly see, does it match, the measured density, the electron three-dimensional image that I've got. And so if nothing else, it's speeding up structural biology tremendously. And as the program becomes more robust and more reliable, for many routine structures, people will calculate the AlphaFold structure and start using it, okay? And I think there are a number of things. One is they need to do the same thing with nucleic acids as what they've done with proteins. That's harder because the database of nucleic acid structures is much smaller. And they are working on protein-protein interactions. For example, complexes of proteins and so on. And if they can, calculate protein-protein interfaces, which they're already doing to some extent, that would be a big major advance. And a third one is if they can look at ligand binding and predict ligand binding with proteins, then you would really transform it, and a lot of what we would have previously done experimentally might eventually be completely done computationally. Although, depending on where the state of the art was, you may want to then check it experimentally.
There's a question at the front here, and then there's also a question up there as well. Oh, there's lots of questions. Sorry.
Just a quick one. As somebody who did crystallography 43 years ago, exposing photographic plates, I feel a bit like a caveman who just went to sleep for 2,000 years and woke up, and I find it impossible to ask you a question, sir. That was a magnificent lecture. Thank you very much.
Right. Can we have the microphone at the front, please? And then also, oh, sorry, you've got the microphone already. So if we take the question from up there first and then here later.
I'm not sure it'll help us with the experimental part, but I don't know enough about how quantum computing can parallelize operations, but it may be possible that it makes calculation, of structures, more powerful. I think I just have to say I don't know enough about the power of quantum computing to know exactly where it would apply and where it wouldn't.
Please.
Thank you, first of all, very much for an excellent lecture. My impression is that these things that you are speaking of are so incredibly complex that it must be very, very easy to get it completely wrong. How often do you have to repeat a measurement of a particular structure before you really think, okay, that's it?
Yeah, so in theory, you're right, but there are a lot of constraints on the structure. For example, we know that the proteins are polymers in which the building blocks have a very specific order of amino acids. That's a very powerful constraint on, so it's not a random collection of atoms. You know, if it was a random collection of 500,000 atoms, I would agree with you, but all of those atoms belong to a polymeric chain, and they can only occur in certain order, and so that puts a very powerful constraint on the structure. And nevertheless, people have actually built the structure incorrectly. For example, they'll get the registry wrong. So what they thought was the 21st residue is actually the 22nd residue, and so on. But there are very good programs which calculate the structure and compare it with the observed intensities and tells you if your structures, your interpreted structure is correct or not. And so people have all these statistical checks for whether their structure is right or not. Now, with electron microscopy, that is becoming more of an issue. The tools for validation are not as powerful yet in electron microscopy. So a number of electron microscopy structures are not as of the same quality as X-ray structures have been, crystallographic structures have been. But that's improving. And of course, now that we have programs like AlphaFold, you can also see if AlphaFold, the predicted AlphaFold structure matches the density correctly. So because of all these checks and balances, we're very confident of that structure. And another thing is, molecular biology is a highly cutthroat field. In fact, I've written a book about the race for the structure of the ribosome, a popular book called Gene Machine, and you'll get an idea of the sociology of science. And the problem is that people will often race towards the same important structure. And whenever this has happened, the structures turn out to be almost identical. And that, again, builds confidence in the technique.
Thank you. We have a question up at the back there.
Thank you. An amazing talk. I'm in the corner. I aspire to teach biochemistry, and I've got a lot of my students here. Could you give them one golden nugget of advice? They.
I don't know. I have to tell you, on the day I got the Nobel Prize, I couldn't get in touch with my wife for many hours because she's one of these people who doesn't use a mobile phone, and she had gone for a walk. And when I finally connected with her, she said, "Wow, I can't believe it. I thought you had to be really smart to get one of those." So, maybe I'm not the right person to give advice. I would say, science is a very uncertain field, but it's a very exciting field. And so you should find something in it that you really excites you and work on that. If you're going to do a PhD, for example. If you're not going to do a PhD, you can use your science background for lots of different things. Everything from, science policy to journalism to analysis, data analysis. It's trained you to think critically. So I wouldn't worry if you're a biochemistry student. You've got lots and lots of opportunities ahead. Try and do something you really find exciting. That would be my advice.
I'm not sure whether it's working. Anyway, I'm not a science graduate. I've specialized in public health. and I was also the Surrey vaccine lead for the COVID vaccinations for the last 20 months. Of course, it was trying to get Surrey residents vaccinated, and I think we managed to achieve that because Surrey now has the highest vaccination rate in the country. What I wanted to know is whenever I used to organize events or I wanted people to come and take the vaccination, people wanted to take Pfizer and said it was related to mRNA, but were not keen on AstraZeneca at all. And I have really struggled trying to popularize AstraZeneca. Now, I'm not a scientist, so I just wanted to know what was the difference for people to have just turned around and said, we don't want it.
Well, I mean, I'm not a vaccine expert, but what we know is that the efficacy of the mRNA vaccines were among the highest of all the vaccines. They were almost sort of 90%, 90 to 95% effective. And against variants, they seem to offer significantly more protection. And so certainly in the Western world, the mRNA vaccines sort of became the vaccines of choice. For example, here, for boosters, the British government almost entirely switched to the mRNA vaccines. Now, the AstraZeneca vaccine had a number of things going for it. One is that it didn't require ultra-cold storage, and therefore, if you were distributing it in developing countries, in tropical countries, it was actually, much easier to do so. And the Oxford group, to their credit, also made a stipulation that for the duration of the pandemic, it had to be sold at cost. So the cost of the AstraZeneca vaccine is much lower than that of the mRNA vaccines. But I think if you're going by simple raw efficacy, and we don't quite understand the reasons for that, one difference, of course, is that the AstraZeneca vaccine was made, the spike protein it makes doesn't have a mutation that stabilizes the pre-fusion form. Now, I don't know if that's the reason or not, but that is one difference. And of course, the difference, another difference is in the way that the protein is made. In the AstraZeneca vaccine, the gene for the protein is introduced into the cells through an adenoviral vector. That piece of DNA then gets translated into mRNA that gets made into the spike protein. In the RNA vaccines, the mRNA for the spike protein is directly introduced into cells. So there are some differences which could result in differences in our immune response, and which could offer possibly different levels of protection, and that could be the reason. But a lot of it is, also once people sort of feel, oh, this one's the one to go for, then that's what they do. So.
Questions there, and then two back after that.
You showed some beautiful pictures and some amazing structures. But what you didn't really explain was why we should be interested in those structures. I mean, what is, as it were, the point of knowing that?
Yeah, I thought I'd done that in the very first slide when I showed a car. Okay, so.
Well, I meant from a medical or biological
point of view. You can imagine, what is the, you have to ask. For example, I showed a structure of a molecule that's a turbine that generates ATP. Now, if you didn't know what that molecule looked like, you would have no idea how it carries out that chemical reaction of taking an ADP molecule and adding an extra phosphate to it. Where does the phosphate bind? Where does the ADP bind? Why did they come together? How is ATP formed? How is it released? So, and how is it all generated by using protons that go from one end of the molecule to another to turn the turbine? You would know none of that without the structure. So that was the point I made with the car, is that if you want to understand how a machine works, you have to know what it looks like. And so it's the same with molecules.
Thank you very much. It was a really interesting talk. One thing that I thought was very striking was seeing how you often had sort of physicists interfering with biology and physics and chemistry into play, and people intruding cross-disciplinary. And I was wondering if you think that's something that is important to have going on, and if so, whether there's anything you think that academics should be doing to encourage that.
My view is that you have all kinds of science. There are fields of science which are not interdisciplinary at all. Well, let me backtrack. They use instruments, and instruments, of course, are made by combining lots of other disciplines. They require engineering, they require physics, and so on. But their own field doesn't actually require something else. Then there are others where they do require other fields. For example, if you're at the cutting edge, there is no instrument, and you have to make your own instruments. Then you have to talk to people who know physics, who know engineering, and so on, and talk to them. Or if you need a particular chemical probe to probe your biological molecule or to light up some part of the cell, you may need to go talk to an organic chemist, who will generate a particular probe for you, or even an inorganic chemist, which would be an extreme step. So the point is that you have to keep your mind open. So what universities should do is to reduce the barriers. You can't force people to be interdisciplinary. It'll happen naturally if it requires it. But if you inhibit it, then it won't happen at all. And so you have to try and reduce the barriers for cooperation between departments. And that I don't know how to encourage that. One is, encourage sort of common meetings. It's nice if they all eat in the same cafeteria, so they bump into each other all the time and talk to each other. And other more informal ways, maybe you could have talks where people from one discipline give general lectures about their work to other disciplines. This happens in Cambridge through the college system. So college systems will have series of talks by fellows. One week it'll be some historian, another week it'll be a molecular biologist, then it'll be some astronomer. So, they get to sort of know each other, and you should maybe encourage that sort of forum.
Thank you. So we've got quite a few questions here, but the microphone's up there. Just before you ask your question, could I stress to people online, there's a question box if you have any questions. If you type your question in there, and then I will receive it here. Please go ahead, sir.
Well, thank you. Thank you very much for the excellent, fascinating talking. And I have one question, because for the vision, you have just like a wavelength to visualize like light. The length is getting certain smaller. What's the resolution to see the things you can have? Obviously, the molecule become smaller and smaller. You want to see as clear as possible, but the wave, the ray sent to the molecule can be very small, but the wavelength could be much larger than the molecule. Do you think what's the limit you can see for the smaller things at the moment?
I mean, what, using light? Well, I mean, it's different. So X-rays typically have a wavelength of 1 angstrom or 0.1 nanometers. And so you could resolve, in principle, things which are about half an angstrom apart. And so if you get to a resolution of, half an angstrom, you're actually almost able to see electron clouds around atoms. So you can get to very, very high resolution in principle with X-rays. Electrons have an even smaller wavelength. And so, in theory, you could get to very high resolution with electrons. What's limiting you is that to get to that high resolution, you would need a lot of signal and you would have radiation damage. So in practice, people have gotten to about 1 angstrom resolution for these large protein molecules with electron microscopy. And with light, light is about 500 nanometers. You could, let's say you'd use blue light or, maybe near ultraviolet light, so you'd say about 350, let's say 400 nanometers. You could then resolve things which are about 200 nanometers in size. Now that's much, much bigger than molecules. I mean, even a ribosome is only about 25 nanometers. But there's a technique called super resolution microscopy, which uses, fluorescent molecules. And then there are very clever ways to look at the peak of the fluorescence and from one floor to another floor. And by using that, they have been able to get the resolution down to about, in the tens of nanometers, certainly. So much, much smaller than the wavelength of the light that they're actually using. But in that case, they're not actually looking at the molecules themselves. They're looking at the distribution of fluorescence from the molecule, okay? So it depends on their labeling, organelles or parts of cells or individual structures with fluorescent reporters that they can then use for this super resolution technique. So it depends a lot. So with super resolution, you can look at molecules that you've coded to about 10 to 20 nanometers resolution. With X-rays, you can go to 0.1 or 0.05 nanometers, similarly for electron microscopes. And with light, you can probably get to 200 nanometers.
Is that a current technology limitation?
Sorry?
Is that a limitation of the current technology?
It's a limitation, but I mean, why would you want to get any, if you're a biochemist, it doesn't buy you a whole lot more if you get to below 1 angstrom. If you get to 0.1 angstroms, you're not going to learn much more.
Winky, thank you for this fascinating talk. So seeing is believing, but I think, related to another question, the whole game is to really to be able to discover and then to make new molecules for drugs and for therapies, right? So my question is on the AlphaFold. So AlphaFold can predict the structure by machine learning, and that to the same extent as you explained, as accurate as the experimentally determined structures.
Right.
But it doesn't solve the problem of the molecular docking.
Very large, for these very large complex structures or, structures inside cells. I think that's still very much an experimental technique. But for individual molecules, you're right,
So, but you still need to, I think there's a lot of work to be done in terms of the molecular docking, in terms of drug discovery. Right?
Yes, although I should tell you that, Demis Hassabis, who, founded DeepMind and really drove the protein folding prediction with AlphaFold, has now started a new company called Isomorphic Labs, where they want to use machine learning to predict drug binding. Okay, and so that if that works, it would tremendously speed up drug discovery.
Thank you for the great lecture. Yeah, so it is very obvious that now we started to accumulate very high resolution structures of a lot of biomolecules, especially with the development of the CRIO-TM and the DeepMind technology. So do you have any kind of a vision on your mind, how it can be translated to kind of a knowledge on the higher order structure, like, I don't know, organelles or cells, which consist of like thousands of molecules?
Yeah, I mean, the trouble with, okay, so when you looked at the stalk when you looked at the stalk of that bacterium, you could see that the stalk surface had a very regular organization, right? But a lot of cells are, a lot of the interior of a cell isn't like that. It's messy, or molecules are moving around. They're not forming regular arrays everywhere. They're sort of coming together and then dissociating. The organelles all look kind of, they don't have a regular shape. They look sort of lumpy and irregular. And so what these molecules do, though, knowing the structure of molecules will allow you to understand how they work themselves and how they interact with other molecules. And that doesn't come just from structure alone. It comes from a lot of different biochemical approaches and genetics to sort of modify residues and test, you know, which residues are important, which are not, what they're doing, etc. So it comes from a combination of approaches from various disciplines in biology and chemistry to really come to that final understanding. Ultimately, what we want to understand is, you know, how does a cell work, you know, with all the sort of thousands of molecules inside a cell? How do these molecules all talk to each other and coordinate processes to do what a cell does, you know, which is to function and, you know, generate, if you're a nerve cell, you have to generate current and, you know, electrical impulses. If you're a liver cell, you have to make lots of enzymes and hormones. If you're an immune cell, you have to make antibodies. So how does all that work in the context of this, you know, orchestra of thousands of molecules? So I think that's sort of the problem in biology. And also, how does a cell develop? How does an organism develop, you know? So those are all complicated questions. And understanding molecules is just one step. But it's a crucial step, but it's one step along the way.
Thank you for the lecture. It was fascinating. I apologize in advance. I am just a humble environmental engineer. Don't know a lot about biology, and this is going to be more of like a philosophical question. Starting with your first, the first slide about the car. My question is there a way of distinguish the tiny little things that you see when you make those structures up, distinguish the car from the driver? I mean, can you see the difference between the molecules that are a machine and the one that drives them? The one that is actually alive and something that's just built up?
Well, I mean, let's say you have a large complex and you have smaller things that come in and activate them. That would be almost the equivalent of the driver. We can do that, and biochemists have actually shown that even before we saw them. But once you see the structures, you can see how this molecule will go and turn on this other molecule, okay? And so, for example, that movie of that dynein walking along the microtubule, that required an activating molecule. Without that activating molecule, the dynein wouldn't do anything, you know? And so, so that sort of thing is very much part of, you know, the broad field of structural biology or cell biology.
So we have one last question down here, please, because we're out of time. Thank you.
Thank you very much for your wonderful session today. My question, I was very much interested in your life journey, starting from physics, doing a PhD in that, and then going and studying from scratch biology. And I was wondering, how was the crossover? What enticed you? Because, you know, I have a physics degree, I have a master's in mass communication, I've got engineering degree. And how do you decide, you know, and in what way the basis of physics helps you? I think it does help in any subject. So what was your inspiration? What was the moment that you decided to go completely different field, and how was the crossover?
Sure. I have to tell you, though, that people often say that, oh, you know, you were able to do this because of your physics background. Well, I can tell you, the two people I shared the Nobel Prize with didn't do a physics degree, you know. One of them was a straight, he went into crystallography and then molecular biology. One person who didn't get the Nobel Prize, Harry Noller, was a straightforward biochemist. So I'm not sure. I think it may have helped me in a subtle way, but not in a very direct way, okay? This is not to say that my physics background was wasted, but I'm just saying, you know, many of my students and postdocs don't have physics degrees, and they're doing just fine, okay? So, so I just want to say, it's not, there's no hard and fast rule. Now, the reason that I left physics to do biology, you can think of a push and pull component. The push component was that I was really bad at physics, you know, and I couldn't, think of a way to really, and I thought if I stayed as a physicist, I'd be some second-rate physicist doing, or well, actually third-rate, doing some boring calculations that nobody would care about. And at the same time, biology seemed to be at a very interesting stage. It actually still is. You know, it's amazing how biology has gone from one revolution to another over the last 100 years. And so, it seemed interesting, and it seemed like it could be done by ordinary mortals, of the sort of intelligence that my wife attributes to me. So, so I felt that
both
that
parents
was a
being
good bet.
PhD as well, so you inherit the PhD within your family as well. I just was looking at, because I was born in Baroda as well, so there's some, you studied there.
So thank you very much. I'm afraid that's all we've got time for. I think Venki's already mentioned that he's written a book, so if you need to know more about the race to the ribosome, there's that. But I did overhear him earlier talking about he's working on another book on an entirely different subject, aging and dying.
Why we age and die, the biology of why
So
we age.
there'll be more from Venki that you can read coming up. Before you go, I have one last thing that I would like to do, which is to say thank you very much indeed for coming and giving us such a wonderful lecture. And as a token of our appreciation, we have this to give you. Thank you. And I think, oh, sorry, this is where we get directed into. Okay. We'll give you the lid. Yeah. So thank you very much for coming, everybody, and safe journey home.